Well, good afternoon. It's a pleasure to welcome you to the 55th Convocation of the Rockefeller University. This afternoon, we will confer doctorates on 17 students. Our convocation is also the occasion on which we recognize individuals for extraordinary contributions to the advancement of science. And today we will confer the degree of Doctor of Science, honoris causa, on 4 distinguished individuals who are esteemed leaders within the scientific community and cherished members of our own university community, Günter Blobel, Paul Greengard, and James and Marilyn Simons. At the first convocation ceremony in 1959, 5 young people were awarded degrees. On that occasion, President Detlev Bronk said, "For those who are to be the leaders and catalysts in the world of creative learning, the quiet intimacy of scholars is required, as is the close personal association between teacher and student." Today's graduates, like their predecessors, have learned science by doing science, side by side with a stellar research faculty. Close scientific collaboration between mentor and student is at the heart of the David Rockefeller Graduate Program. That association will be evident to you now, as each student is presented personally by a member of our faculty.
Good afternoon and welcome, Mr. President, Mr. Carson, graduates, Rockefeller community, and honored guest. We have a unique tradition at Rockefeller in which we focus on the graduate. I now give my formal assurance to the President and to the Board that these graduates have satisfied their obligations and are eligible to be conferred the degree of Doctor of Philosophy.
Good afternoon, everyone. As I stand here before you, I feel a real sense of pride in Pinar Ayata's achievements. This young woman has so many facets to her personality and such strong passions for nature and the world about us that at first I thought she'd have difficulty finding the balance between science and society that all of us must achieve to succeed in this endeavor. Of course, my concern was completely unnecessary. Pinar has been blessed with the boundless energy and enthusiasm to embrace each day as a new experience, with a deep intelligence to guide her with clarity towards her scientific goals, and with an unfailing optimism to carry her through the storms of scientific research. These qualities have allowed her to make startling discoveries in the field of epigenetics and Rett syndrome, while at the same time scouring the world for remote mountains to climb, snowfields to traverse, and rivers to kayak. Rockefeller has served Pinar very well. She has developed a talent for science and life that eludes many of us for many decades. In 2009, my laboratory discovered that mammalian genomes contain a 6th nucleotide, 5-hydroxymethylcytosine, which is particularly abundant in the brain. This ignited a fire in epigenetics research that continues unabated today and has led to 250 papers in the last few years. Yet as interesting as these many papers have been, it is Pinar's discovery of the mechanism for decoding 5-HMC in the nervous system that has shed most light on this new process for genome regulation. Through a series of beautifully executed biochemical experiments, Pinar discovered and proved definitively that the protein that reads this new mark in the genome is encoded by the gene that is causative for Rett syndrome, a devastating autism spectrum disorder that afflicts young girls. Now, I found this result shocking because the protein had been known and studied for a couple of decades, and because two major laboratories had published that it does not bind 5-HMC. So I bet Pinar that something must be amiss, that there has to be another protein interpreting this new genomic signal in the brain. Well, with the exacting experimental skill that is typical of Pinar, she proved me wrong, publishing her results in a landmark Cell paper and winning a bottle of champagne that I must admit I haven't paid her yet. As I hope you can appreciate from these comments, Pinar's childhood in Istanbul, her early experiences sailing the Mediterranean with her family and friends, her educational experiences at Sabancı University, and her scientific internships in Heidelberg and Boston seem to have primed her for scientific and personal explorations that stretch the imagination. I've been fortunate to watch as these excursions into the unknown bore fruit, and I am confident that they will continue to do so during her postdoctoral studies at Mount Sinai Medical School in the laboratory of Dr. Ann Schaefer. Mr. President, Mr. Carson, and honored guests, it's my pleasure to present to you Pinar Ayata.
Nicole Bowles graduated from New York University in May 2008 with a Bachelor of Science in Chemistry and a minor in Math. Prior to coming to Rockefeller, Nicole had worked as a summer researcher at the University of Pennsylvania and then at the Joslin Diabetes Center, and the next year at Merck Pharmaceuticals, and finally, after graduating from NYU, at the Mount Sinai Department of Pharmacology, studying the endocannabinoids. Endocannabinoids are the body's own chemicals that are the reason it responds to marijuana. And they are increasingly recognized as having many important functions in the brain and throughout the body. A parallel example, endogenous opioids are the reason the body responds to pain-killing and addictive opioids. Doctors Matt Hill and Ilya Karatsarios were postdoctoral fellows in our lab at the time that Nicole joined us, and they had interests in endocannabinoids and circadian clocks, respectively. This was part of our ongoing work on stress effects on the brain and body. Nicole officially joined our lab in the spring of 2009, along with her past work and interests and the advice from Hill and Karatsarios. This led to her thesis, which was entitled Cannabinoid CB1R Receptor Mediates Metabolic Syndrome in Models of Circadian and Glucocorticoid Dysregulation. So what does this mean? Our stress hormones and the body's own biological clocks play a key role in metabolism and the regulation of body weight. And Nicole's thesis addressed how the stress hormone, cortisol, and disruption of the normal daily biological rhythm regulated by those clocks contribute to obesity and the metabolic syndrome. And what she discovered is that mice lacking this CB1 receptor were protected against all these changes in metabolic function. More importantly, blocking the CB1 receptor not only globally with a drug that acts throughout the body, but also through a drug that does not enter the brain and only works peripherally, also was effective in preventing these effects. In addition to her ambitious and successful laboratory work for her Ph.D. thesis, which Nicole managed with typical efficiency, she also volunteers in a diverse array of community activities in the U.S. and abroad that are directed to helping overcome the effects of poverty, social inequality, and improving education. Upon coming to Rockefeller, Nicole joined and soon became co-chair of our student Pugwash, which examines the relationship between science and society and the common goal of ensuring that scientific research benefits humanity. She also applied this interest directly by volunteering to help individuals and families in her own neighborhoods of Philadelphia and New York, for example, through Meals on Wheels and events sponsored by New York Cares, including weekly tutoring in a public high school and preparation work for soup kitchens. And she's also used winter and spring breaks for Habitat for Humanity, week-long construction projects in Virginia and North Carolina, along with a trip to New Orleans for continued hurricane relief efforts. And she used her 2-week 2011 vacation to travel to Peru and work on childhood education. Clearly, Nicole is not only an excellent scientist, but has an incredible social conscience and puts this into practice. Her next steps will be to continue in our laboratory as a postdoctoral fellow while pursuing a master's degree in epidemiology and public health across 68th Street at Weill Medical College. Mr. President, Mr. Carson, and honored guests, it's my great pleasure to present to you Nicole Bowles.
Fabio Casadio came to my lab from Italy, which is relevant, but he came with carrying an interest in what turned out to be the centerpiece of his research work in my lab, which was a topic called arginine methylation. I say the Italy's relevant because I'd like to welcome his parents that are here from Italy, and certainly I know they're proud of him, but we are proud of him as well. Now back to arginine methylation. I'll keep it simple. I'll just say that in the current news of our day, this is the little brother of a big brother, lysine methylation, that seems to be really sort of guiding our genome and, in fact, watching out for the arginine methylation below it. And that makes it very rare. It makes it technically challenging, but Fabio actually sort of cracked that open and in fact, taught us quite a bit about this little brother. He taught us who puts this mark on, who takes this mark off, and most importantly, how it functions in the cell. Fabio mapped a site that in terms of the structure that we study in the lab is so critically positioned to be a genetic switch that might open or close our genetic material. Fabio put forward the hypothesis that that may indeed be what this modification is all about, and went on to test this in a defined chromatin system that took advantage of work with Bob Rader and Tom Muir. Now, relevant at a personal level is Fabio wasn't just smart upstairs. He was quick. He was actually quick downstairs, meaning his feet. Fabio is a very big sports man, and in fact, of the many sports that he did while he was here, he regularly entered, I think, all the premier marathons. But maybe more surprising to me was that, in fact, he came in as the elite marathon top-tier runner in all of those races, and I know a lot of Rockefeller students dominate, but Fabio's even a notch above. So what's he going to do with all this stamina and discipline? He's decided to take another course. He's going to take his smarts and quick wits to McKinsey & Company and work there. And we know that he's got all of the background ingredients for this because he can be so disciplined and has the stamina and the strength. But in fact, I learned recently when Fabio graduated that he has been turned on to one American delicacy. This is actually a calorie-packed food. It's actually cheap, and that makes it graduate student friendly. And it turns out to be nothing other than peanut butter. So Fabio loves peanut butter. I mean, it's kind of weird what you learn about your students. So I actually have an added present for Fabio's parents. I've already gotten Fabio a big jar of peanut butter, but I'd like him to take this back to his parents because they've traveled so far. This may help them on the return trip. So Mr. President, Mr. Carson, and honored guests, it's my absolute pleasure to introduce to you Fabio Casadio. And I learned some Italian. Bravo, Fabio.
Emily Kahn Gantman came to Rockefeller as an extraordinarily promising young talent and has emerged as an outstanding multidiscipline scientist, a humanist, and, among other things, a mother. A lot to get done during the course of her time here, both a Ph.D. and producing a new baby. She came to Rockefeller very accomplished from the University of Pennsylvania. She had a 3.9 GPA, a wide range of research experiences. It included a bunch of research that she had done. She had done clinical research at CHOP, basic science in Arabidopsis, plant system, and work that earned her the Rose Undergraduate Research Award at Penn. And she was a true scholar, which presaged in many ways her time at Rockefeller. She got an A+ in her last year in a range of courses, including computational biology, which she ended up using in her thesis extensively, and physics, which I have to admit she did not use in our lab. She followed Penn with a one-year fellowship then at the NIH and struck a balance there between clinical and very basic science, doing medical research on developmental biology. And that brought her to Rockefeller with an intense interest in the molecular mechanisms of disease. So she carried her erudition here. She earned honors in her coursework, something I didn't know we even handed out. And I have to admit, even my father, known to be a bit reserved for his praise, called her term paper an imaginative success. Wherever he is, there he is. And, her interests were a natural fit for our laboratory, so she came here and began a series of extremely ambitious and creative experiments, and that earned her a Women in Science Fellowship at the Rockefeller here. The range of work that she accomplished is breathtaking, and I would almost say embarrassing to me. The amount of work that I let her approach was enormous, but Emily was really unstoppable. She was the first and really to date only graduate student in our laboratory who has done and embraced really both the clinical side of the laboratory as well as the basic research side. And so in a sense she developed two theses in the course of one, falling in love originally with clinical questions about what drives anti-tumor immunity in patients with rare clinical diseases called paraneoplastic neurologic disorders, in which they can eradicate their cancers, even while becoming more attracted over time to basic questions about the mechanisms of what drove the activation of such T cells. So it was this work that really drove the bulk of Emily's thesis. She had an outstanding ability to project her own limitless enthusiasm and delight for science onto others, which ended up first intellectually engaging them and then charming them into working collaboratively with her. This led to an extraordinarily complex thesis, one of the more complicated ones any student in my lab has produced, genuinely going where nobody has gone before to look at very detailed aspects of the regulation of the dark matter of the genome, that part that involves the regulation of RNA by RNA binding proteins and other things such as microRNAs, and looking specifically in resting versus activated T cells. She produced a copious amount of extremely interesting work that I truly believe will be recognized as a foundational study in the years to come. Emily's ability to gather support from her colleagues, I point out because it turned out to be especially useful after one particular lab meeting. At the end of a detailed hour going over data that was buried inside her head, she showed on her last slide a fuzzy black and white picture that turned out to be an ultrasound of what was going on in her uterus. And this, in fact, ultrasound turned out to be a wonderful means of aggregating additional help from the lab, as nobody would let her within 100 feet of an isotope. And she, in fact, ended up redoubling her collaborations in the group. So Emily is now a happy mother. She's looking forward to coming back to the lab for a year's postdoc as she plans her future as a true woman in science. Mr. President, trustees, Mr. Carson, it's my pleasure to present Emily Kahn-Gantman for the Ph.D.
I will read remarks prepared by Tizia DeLonga, who unfortunately couldn't be here today. It's difficult to do justice to Teresa Davoli's remarkable talent in the 2 minutes allotted to each graduating student. Teresa left campus about a year ago to join Steve Elledge's lab at Harvard, where she is supported by a Helen Hay Whitney Fellowship. Here at Rockefeller, she was honored with the David Rockefeller Fellowship, the Anderson Center Fellowship, and a Woman in Science Fellowship. Teresa joined our lab in early 2008, at a time when she was still speaking her charming, but at times challenging, staccato Italian English. She is from Parma and became enormously popular because she would bring us her father's wine and amazingly large chunks of the best Parmesan cheese you can imagine. Teresa joined the lab because she had a keen interest in cancer research, and she had already done work in this area with Pier Giuseppe Pellice in Milan. To explore a cancer-relevant aspect of telomeres, she studied how the loss of telomere function contributes to genomic instability in cancer. What followed was a breathtaking period in which Teresa ran her own independent research project and demonstrated that shortening of telomeres can drive cells to duplicate their whole genome. Because telomeres shorten in the early stage of tumor development, Teresa's observations help explain why so many cancers have twice the normal number of chromosomes. Earlier this year, Teresa was awarded the prestigious Hal Weintraub Award for her work. Hal Weintraub was a brilliant scientist and a much-loved mentor. Fred Cross, a member of Teresa's thesis committee, was a postdoc with Hal. He wrote to Teresa when she received the award as follows, and I quote, "Rereading your telomere paper, I was struck with the depth and sophistication of the analysis. Not just DNA damages causes overreplication, but a whole detective story, from telomere shortening in hyperplastic cells to polyploidization and the possibility of aneuploid disasters in cells that survive crisis. Hal would have greatly appreciated your work," end quote. Mr. President, Mr. Carson, and distinguished guests, it's my great pleasure to present to you Teresa Davoli.
Good afternoon, and congratulations to the graduates and their families. It's my pleasure to present Paul Dosa. Humans are constantly exposed to bacteria, and the colonization of our tissues with beneficial microbes after birth can help our metabolism and shape our immune system. But unfortunately, we occasionally encounter harmful bacteria, and that usually causes deadly diseases. And as many of you know from the general press and other organizations, we are running out of ways to treat bacterial infections. Currently, due to the risk developing antibiotic resistance to currently available antibiotics, and a limited pipeline of new drugs that are emerging. So my laboratory has been interested in developing small molecules that would selectively target bacterial pathogens over helpful bacteria. So towards this ambitious goal, Paul Dosa was the first graduate student in my laboratory to embark on these studies. And Paul's thesis work really allowed us to discover and characterize drug-like molecules from medicinal plants that could selectively target bacterial pathogens. And they've served as the basis for a set of new anti-infective molecules from the lab. In addition to those sort of initial studies, Paul's thesis work also helped to characterize the mechanism of action for additional synthetic molecules that have been developed. And his thesis work has really paved the important foundation for my lab for the development of new anti-infective strategies in the future. So for these studies, I would like to thank Paul for his important contributions to the lab and wish him the best in his future. And Mr. President, Mr. Carson, honored guests, it's my pleasure to present you Paul Dosa.
It's my pleasure to introduce to you Amy Grunbeck. Before coming to New York, Amy received her BS degree with honors from Dickinson College in Carlisle, Pennsylvania, where she won the American Institute of Chemists Award, which goes to the highest ranked chemistry student each year. Dickinson is a small liberal arts college, but it has a fabulous undergraduate chemistry program. Some of you may not know about Dickinson, but it has a very interesting and relevant history. Dickinson was founded in 1783 by Dr. Benjamin Rush, who was both a physician and a professor of chemistry, not a bad role model, as well as a signer of the Declaration of Independence. Rush envisioned a great college at the edge of the frontier, and central Pennsylvania was at the edge of the frontier in those days, where both men and women could receive a science-based progressive education. Dickinson remains one of the country's best small colleges. Well prepared as a chemist at Dickinson, Amy did spectacularly well in her thesis research on a very challenging and important question in pharmacology as it relates to drug discovery. She tried to understand with chemical precision how an important class of membrane receptors called heptahelical receptors transmits signals across membranes. Amy was able to achieve what I feel is a major breakthrough in the field. She applied a new technology called amber codon suppression to introduce a unique type of amino acid that is normally not found in nature into receptors of interest. The amino acid she chose could be coaxed to react with neighboring molecules when exposed to strong ultraviolet light. She used this method to map the interactions between receptors and therapeutic drugs, whose sites of binding and modes of action were not previously known. The strategy is especially powerful because it can be done in cells where the cross-linker amino acids are genetically encoded into the target of interest, in this case into a receptor for a therapeutic drug. Amy's method is now widely termed targeted photocross-linking, and it is very cool to chemical biologists. Amy's work has received significant attention and praise. She has been invited to give platform talks at Banff, Alberta, Canada, in San Diego, and at an important receptor pharmacology meeting in Brazil. She has also been featured in a Meet the Author interview for a prominent American Chemical Society journal. And in fact, she just shot another video interview 2 days ago. Amy's published research forms a substantial, coherent body of work that I predict will be taught in chemical biology textbooks in the future. And on a personal level, Amy has been a pleasure to work with. She has been perhaps the most consistently upbeat and team-oriented member of our group over the past 5 years, a uniquely talented and likable person. As she moves to Boston, we wish Amy all the best in her future scientific career, and we're very proud of her accomplishments. President Tessier-Lavigne, Mr. Carson, honored guests, it is my privilege to present to you Amy Grunbeck. It's now my pleasure to introduce to you Adam Knapp. Adam's thesis research focused on how a particular class of cell surface receptors, called G-protein coupled receptors, assemble in the cell membrane with other receptors to form a molecular nanomachine called a signalosome. He used a combination of computational and experimental approaches to model the disc membrane of the rod cell in the retina at the back of the eye. Adam showed that the light receptor called rhodopsin first forms pairs, or dimers, that then assemble into long rows. The rows then line up side by side, forming a type of surface structure that resembles a toy slot car track. The slot between the dimer rows contains lipids from the membrane bilayer. Transducer proteins inside of the cell can hold on to the track and slide along the slot, using their own lipid tails as they carry out a one-dimensional search for a rhodopsin that was activated by light. And only about 1 in 100 rhodopsins are active at any one time, even in bright light. For a fast visual response, the transducer has to find its receptor as quickly as possible. Once the transducer finds a receptor, it bounces out of the track to interact with an enzyme in the cell. So imagine a protein sliding along a molecular slot car track on the membrane surface and getting knocked off the track once it hits an active receptor, a protein bump in the road. Adam's very cool model in part explains the fast physiological response to light and vision. Adam also developed a useful enabling technology to measure whether GPCRs are properly folded and how stable they are. Adam's method attracted the attention of biotech and pharma companies, in part because his relatively low-tech but clever method needed only about 1/10,000th of the amount of material that they had been using in their own very expensive biophysical assays to measure basically the same thing. One company currently using Adam's technology was very surprised when they found out that Adam himself could produce the same amount of data in 1 week that it took 3 of their own technicians 1 month to produce. Adam's innovative thesis work will continue to pay dividends to future lab members as his methods are more widely applied. Adam received his BS in chemistry from Stanford. He was a member of the so-called Facebook class. Do any of you know what the Facebook class is? I first came to know of that term in a New York Times article about 2 years ago. In short, the Facebook class at Stanford was the first class that was given exclusive free access to the Facebook social media application that was being developed just up the road by Mark Zuckerberg after he moved west from Harvard. When I asked Adam about the Facebook class, he said it was no big deal. Oh, but by the way, he has something like 900 Facebook friends, which is absolutely insane. Adam is the type of person who never seems to have a bad day. He's an enthusiastic problem solver and always seems to rise to the occasion. For example, is when I asked him to fly to Germany with 1 day's notice to help with a major presentation at a very contentious meeting. Adam is one of the few people I know who has traveled more than I have in the past 4 years, with carefully planned adventure trips to Kilimanjaro, Machu Picchu, and Southeast Asia. And he also attended many Stanford Cardinals football games, including key bowl games during the Andrew Luck era. Adam will start at McKinsey & Company this summer, where his enthusiasm and skills will be put to very good use, I'm sure. President Tessier-Lavigne, Mr. Carson, and honored guests, it is my privilege to present to you Adam Knapp.
I will read remarks prepared by Jim Hudspeth, who unfortunately couldn't be here today. One important aspect of hearing is recognizing the sources of sounds in our environment. In nature, there is an obvious selective advantage in distinguishing a prospective meal, perhaps a tasty pig rooting in the bush, from a predator such as a lion. With human speech came the necessity to differentiate voices, which we can still do, even in a crowded environment. Still greater refinement underlies our capacity to make distinction between the various instruments in an orchestra. We can distinguish different sounds by their content of particular frequencies. A female voice is higher than a male voice. A violin is pitched above a viola. These discriminations commence in the cochlea of the inner ear, which functions like an inverse piano to decompose complex sounds into their tonal constituents. Each sensory cell in the cochlea is tuned to a specific frequency and sends signals about that frequency along nerve fibers into the brain. Investigating the flow of information from the sensory cells to the nerves, Suchit found that this interaction is also tuned to specific frequencies. In other words, the chemical synapses at which sensory cells communicate with nerve fibers and themselves help us discriminate sounds. This result, which represents the first demonstration of tuned synapses, will encourage a search for additional examples of the phenomena in the nervous system. Suchit has an interest in clinical medicine as well as research. He earlier worked on several medical problems and encouraged disadvantaged high school students to enter medical careers. He has served as executive director of the Weill Cornell Community Clinic, a voluntary organization that provides medical services to the indigent, and founded the Heart to Heart program that offers citywide screening for cardiac problems. As a student in the MD Ph.D. program, he will now complete his clinical work. Thereafter, Suchit plans to enter into a clinical career in which his personal warmth and intellectual acuity will serve him well. Mr. President, Mr. Carson, and distinguished guests, it is my great pleasure to present to you Suchit Patel.
A child that gets a group A streptococcal infection that is adequately treated will not suffer any major consequences. However, a pharyngitis or strep throat from the same organism could result in rheumatic fever and rheumatic heart disease. Although intensive research into the cause of rheumatic fever has gone on for decades, a clear picture as to the connection between the throat infection and heart damage is still unclear. One of the research tools used to examine the infection process of the streptococcus are human pharyngeal cells in culture, the only cell line available from the human oral cavity. These cultured cells have been used for decades to examine the attachment and invasion of streptococci and the genes responsible for this event. However, the streptococci do not infect the pharyngeal cells. They specifically target the tonsillar cells. When Dennis Spencer joined my lab, he wanted to examine streptococcal interactions with tonsillar cells to see if they differed from the pharyngeal cells. He found that when he used human tonsillar tissue discarded from surgical procedures, the variability in his results were such that clear decisions could not be made. However, like much of science, a bit of luck came his way. In his reading, he discovered a paper in which a surgical group reporting on head and neck cancers identified two cancers in human tonsils. Dennis contacted the authors and received samples of these cells and began to extensively characterize and compare them to the pharyngeal cell line. What he discovered was that they were good representatives of the tonsillar cells because they had not changed as a result of the cancer. More importantly, the streptococci were able to attach and invade these tonsillar cells 10 times better than the pharyngeal cells. Furthermore, when he examined the streptococcal genes that were turned on during this interaction, he found several candidates that had not been identified before. We believe that the tonsillar cell line will soon replace the pharyngeal cell line for further studies on streptococcal infections, and in doing so may point the way to finally understand the connection between the throat and the heart. Of all the students and postdocs I've had over the years, I must say Dennis is by far the best dressed and loves to wear bow ties at special occasions, but I don't think he's wearing one today. Not special enough, I guess. On a more serious note, Dennis devotes a lot of his time to mentoring students, not only in the lab, but also on a national level with his 8-year affiliation with the Student National Medical Association. Dennis is now very busy doing his clinical studies at Cornell Medical School. In fact, they gave him the day off today so he could graduate. And I'm happy to report that his experience in the lab has turned his attention to ear, nose, and throat as his medical specialty. Mr. President, Mr. Carson, honored guests, it is my pleasure to introduce Dennis Spencer for Doctor of Philosophy.
So Chan Lek Tan is a brave young traveler on a historic path to conquer the wild and woolly West. Born in Malaysia, Chan's first Western migration brought him to the United Kingdom, where he graduated with honors from Cambridge University with a Bachelor of Arts degree in natural sciences. Inspired by his success, he continued his journey towards the West and entered the Ph.D. program at Rockefeller University. Determined to venture even deeper into unknown territories, he joined my laboratory to work on the role of microRNAs in controlling brain function. Chen's research showed that even the tiniest pieces of RNA can have a huge impact on brain function. In a set of very elegant genetic, molecular, and behavioral experiments, Chen identified a 25 nucleotide long microRNA that controls neuronal excitability and functions as a key regulator of motor activity. Even more dramatically, it has a major effect on seizure susceptibility in mice. Chen's education at Cambridge University gave him not only a sense of irony that he would generously share with his friends and colleagues, but also seeded a deep passion for literature. In order to satisfy Chen's appetite for books, and without compromising his devotion to his experiments, he became a true devotee of audiobooks. These audiobooks would cause Chen to erupt occasionally with seemingly inexplicable laughter in the middle of the day while pipetting at his bench. People were a bit worried about him until we explained what was going on. In his free time, Chen likes to travel, he enjoys good food, and is a great listener and a very promising, talented young man. Chen will now continue his tireless quest westward by pursuing the great unknowns of neuroscience in the laboratory of Zach Knight at UC San Francisco. As he continues his trip around the world, we wish Chen all the best for his next adventure. Mr. President, Mr. Carson, and honored guests, it is my pleasure to present to you Chen Lak-Tan.
Hello again. Sarah Van Dresch came to our laboratory as a hockey player as well as a scientist, and she consistently has managed to slap the puck around the lab, scored an amazing goal during her thesis work here. Sarah is one of the most ingenious, careful, and very committed scientists that we've seen in our laboratory. She came here from Wellesley College with high grades, great GREs, a perfect 900 on her biology advanced subject test, and the Fiske Prize in biology. The latter was for a series of experiments she had done, progressing in interest from anatomy and physiology to more detailed functional studies at the molecular level. In fact, Sarah described herself and her view of the beauty of cellular machinery in her application to Rockefeller as intricate, eloquent, biologically cool, as if inspired by Rube Goldberg and Dr. Seuss. Okay, well, her mentors described her in somewhat more conventional terms, but no less praiseworthy, as a gem with the best hands they had ever seen. We were excited to get Sarah in the laboratory, and her work here has been characterized by extreme focus and attention to detail. This can be seen in looking back over her original thesis proposal, in which she planned to validate the FMRP Fragile X target transcripts by clip, something I'll explain in a second, and then analyze them in specific cell types. And in fact, this is exactly what she did, which is essentially unheard of. Virtually nobody in my lab projects something and actually carries it out. But Sarah did this and stuck to her plans over the years. And she did so with a wisdom that produced, together with her co-thesis advisor and mentor, Jennifer Darnell. Jennifer is there. Raise your hand, Jen. There we go. Producing what I consider to be one of the most important discoveries our lab has made. To explain it very briefly, the commonest form of inherited intellectual disability, which is often coupled with autism, is the Fragile X syndrome. As opposed to most such difficulties in childhood, the genetic cause of this disorder has been known since 1991, over 20 years. Now, in a sobering reminder of how difficult science is, although the gene was cloned so long ago, in all that time there's been no clear understanding as to what the Fragile X protein actually does, what its relationship is to autism, and therefore there's been no clear logical window in how to approach these problems clinically. Sarah and Jennifer worked side by side to execute a true breakthrough piece of work. They identified what the protein did for the first time. They found that it bound to specific mRNA molecules that code for certain synaptic proteins and certain other proteins that are important epigenetic regulators of the sort David Alice works on, triggering a collaboration that's ongoing between his lab and ours now. They also went on to find that this protein, the way that it works is that it arrests translation by ribosomes. Now, Sarah deserves the highest of gold stars for sticking with an enormously difficult experimental problem. She showed basically that the Fragile X protein blocked translation by forcing the translating ribosomes to stop dead in their tracks. So this work was published in the highest of journals, Cell, and has provided the gold standard for our understanding of how the Fragile X protein works and offers great insights into possible disease treatments. Moreover, it led to an exciting and unexpected link between the Fragile X syndrome and autism, and most recently is giving insights amazingly into schizophrenia. All of these avenues are being pursued by our laboratory and others and provide a testimony to the extremely special nature of Sarah's work. Both Rube Goldberg and Dr. Seuss would be proud, as I know her whole family is, and although this is not a prerequisite for graduating from my laboratory, her family includes a new baby. So our lab is certainly extremely proud of her, and Dr. President, Mr. Carson, trustees, I'm very pleased to present Sarah Van Dresch for the degree of Ph.D.
Professor Donald Pfaff had an obligation that prevents him from being here, but as a member of Amy Wells Quinkert's thesis committee and also a neighbor on the same floor of the Weiss Tower, I'm very delighted to do the honors. Don Pfaff writes, "The most fundamental and powerful function of the brain is called generalized central nervous system arousal, essential for consciousness and for the initiation of all human behaviors. Traumatic brain injuries by car accidents or war injuries, for example, are devastating because they damage generalized arousal pathways in the brain. Amy Wells Quinkert has addressed these arousal mechanisms during her years at Rockefeller. Just before she began her thesis work, Dr. Nicholas Schiff, across the street in the neurology department at Cornell, startled everybody by using deep brain stimulation to provoke the recovery of consciousness of a patient who had been in a vegetative state. Amy, in work that was for the first of its kind, demonstrated that the temporal patterning of such deep brain stimulation in laboratory animals could regulate stimulation effectiveness, even when all other physical characteristics of the stimulus pulses were held constant. Her most effective brain sites were in the central thalamus, amongst neurons whose axons project widely to the cerebral cortex. Further, her deep brain stimulation had the effect of changing behavioral patterns from those typical of animals' sleepy time of day to those typical of the animal's normally active time of day. Thus, Amy could deal with a deep global brain function in a precise and quantitative manner. Preparing for Amy's graduation, I was surprised, as Don wrote, to hear that she has a hobby, knitting. Surprised because as a typical Rockefeller professor, I thought I had successfully reduced her spare time to zero. In the race for the Ph.D., Amy has finished ahead of her husband, who works in Brian Chait's laboratory, and when he is ready, they'll head for postdoctoral research in Boston. Mr. President, Mr. Carson, and honored guests, on behalf of Don Pfaff and also myself, it's a great pleasure to present to you Amy Wells Quinkert.
Corey Bargman was unable to be here today, and I'm extremely pleased to be able to present Laura Winston Reed on her behalf. To me, Laura has always embodied the spirit of Dorothy from The Wizard of Oz. She hails from Edmond, Oklahoma, and moved to Lawrence, Kansas to do her undergraduate work at the University of Kansas. There, she was a Phi Beta Kappa Scholar of Genetics, a fly researcher, as well as a proud member of the sorority Kappa Alpha Theta. In 2005, she was magically picked up and deposited at the gates of the Emerald City at 66th Street and York Avenue, an exotic Midwesterner, Jayhawks fan, sorority girl. In Corey's lab, Laura carried out a genetic screen to dissect the rules of synaptic organization in the worm. Using a fluorescent marker to tag synapses, the point at which two neurons communicate with each other, Laura screened for worms with aberrant synaptic morphology. This involved many hundreds of hours looking for tiny faint spots along a worm neuron that changed in a potential mutant. She succeeded in identifying mutations in a gene called synaptotagmin as responsible for defects in one of her mutants. Laura was an enthusiastic supporter of our graduate program, always involved in recruiting students at the Spring Open House, and in one very memorable trip back to Kansas, she accompanied me on a seminar trip to pitch our graduate program to UK biology majors. Somehow between her community service at Rockefeller and her intense worm gazing in Corey's lab, she found time to appear and earn $2,300 on a TV game show called Cash Cab. This particular Dorothy will not be going back to Kansas, but plans to stay in our fair Emerald City to pursue a career in management consulting with the eventual goal of applying her prodigious scientific intellect and her magical social skills to equity research in the biotech sector. Mr. President, Mr. Carson, and honored guests, it is my pleasure, on behalf of Corey Bargman, to present to you Laura Winston Reed.
Graduates, please rise. By the authority of the Regents of the University of the State of New York, which they have vested in the trustees of the Rockefeller University, and is by them delegated to me, I confer upon you the degree of Doctor of Philosophy, which admits you to all the rights and privileges thereunto pertaining. Let us turn our attention to today's recipients of the Doctor of Science honoris causa. This year, we honor 4 great champions in the scientific community: Günter Blobel, Paul Greengard, and James and Marilyn Simons. Leaders, visionaries, mentors, role models, magnanimous benefactors, all these apply to our honorees. All have had an enormous impact on science, both globally and on our own Rockefeller University campus. It is, therefore, a great privilege to recognize them today. I will introduce Paul first, then Marilyn and Jim, followed by Gunter. Each of them will give remarks. Paul Greengard is one of the pioneering neurobiologists of the 20th and 21st centuries. In the 1960s, when most neuroscientists were concentrating almost exclusively on the role of electrical impulses in the brain, Paul chose to focus on the brain's chemistry instead. Thus began a series of historic experiments that have helped to explain the roles of molecules such as dopamine and serotonin in shaping our thoughts, our emotions, and our memories. Paul's influential discoveries, which were recognized by the 2000 Nobel Prize in Physiology or Medicine, have transformed our understanding of neurological and psychiatric disorders. His insights have changed the way we think about treating such diseases as Alzheimer's disease, Parkinson's, schizophrenia, and depression. His sophisticated application of classical biochemistry to the study of the brain has inspired generations of neuroscientists. Paul was born and raised in New York City. After attending public schools in Brooklyn and Queens, he served the nation during the Second World War. He spent 3 years in the Navy as an electronics technician, working with a team at the Massachusetts Institute of Technology to develop an early warning system designed to intercept warplanes before they could reach the ships of America's Pacific Fleet. After the war, Paul attended Hamilton College in upstate New York, where he graduated as a math and physics major in 1948. He considered graduate study in theoretical physics, but changed course because the Atomic Energy Commission was the only source of fellowship support in that field at the time. Paul decided instead on biophysics, and this was where his career trajectory began to intersect with the Rockefeller University. He commenced his doctoral studies with Detlef Bronk, the future Rockefeller president, at the University of Pennsylvania. When Bronk left Penn to become the president of Johns Hopkins University, he invited Paul and several others in his lab to establish a new department of biophysics at Hopkins. It was there that Paul met future Rockefeller scientist and Nobel laureate H. Keffer Hartline, who became his first research mentor. Paul's thesis advisor at Hopkins, Frank Brink Jr., was brought to Rockefeller by Bronk and helped shape our graduate program. After receiving his doctorate from Johns Hopkins in 1953, Paul conducted research at the University of London, Cambridge University, and the University of Amsterdam. He returned to the United States, where he worked at the NIH before becoming director of the Department of Biochemistry at the Geigy Research Laboratories. He joined the faculty of Yale University in 1969. It was at Yale that Paul began transformative research on communication between nerve cells. In the ensuing years, he provided the framework for understanding how the nervous system functions at the molecular level, setting the stage for important work that is now underway in laboratories around the world. To cite just one example, in the early 1970s, Paul and his colleagues identified enzyme activity in the brain that was triggered by the neurotransmitter dopamine. This led to the discovery of the dopamine receptor. The mechanisms that Paul and his colleagues revealed are essential to contemporary research on the origins of addiction, attention deficit hyperactivity disorder, mood disorders, schizophrenia, and movement disorders such as Parkinson's disease. In 1983, Paul finally came home to New York, joining the Rockefeller University to establish a new laboratory that has taken his resilient concepts and applied them in multiple areas of neuroscience. Since 1994, he's also headed the university's Fisher Center for Alzheimer's disease research. His laboratory's many accomplishments include showing that dopamine's binding to its receptor on the cell surface triggers a cascade of reactions that in turn regulate a variety of functions in the cell. These include the activation of an enzyme that chemically modifies other proteins by adding phosphate groups to them. Paul's laboratory also discovered a central signaling switch, a protein known as DARP32, which is influenced by dopamine and other neurotransmitters. Paul shared the 2000 Nobel Prize in Physiology or Medicine with Arvid Carlsson and Eric Kandel. Paul donated his monetary share of the Nobel Prize, and together with his wife Ursula von Riedingsvard, established the Perlmeister Greengard Prize to honor his mother, who died while giving birth to him. The prize, awarded in her name, shines a bright light on the accomplishments of women scientists. This year marks the 10th anniversary of the prize, which has been given to 14 women scientists, with the hope to increase the likelihood that women will receive their fair share of the highest honors in science. All of us at Rockefeller are profoundly grateful for all that Paul has contributed to science, to the scientific community, to women in science, and to Rockefeller, and we're delighted to have this opportunity to honor him. Paul, please rise to receive your hood and diploma.
Mr. President, Chairman Carson, members of the Board of Trustees, and members of the Honorary Degrees Committee. Before making my presentation, I'd like to mention, some of you may have noticed my hat kept falling off my head. Being a scientist, I came up with a hypothesis. It was because I don't have any hair on my head. So there's no friction. And then I looked around at all the other faculty, and they all had hair on their heads, and they all had a hat on. And then I looked at Professor Günter Blobel. And being a critical scientist, I rejected my hypothesis. I've been meditating on what might be the reason that he and I are the only ones without hats on our heads. Thank you, Mark, for your wonderful and gracious comments. I feel deeply pleased to be a recipient of an honorary degree from the Rockefeller University, and I wish to express my deep appreciation to you and to the members of the Honorary Degree Committee for this recognition. I'm also delighted to be sharing this recognition with three individuals who I so much admire, namely Jim Simons, Marilyn Simons, and the gentleman without a hat. It is customary for honorary degree recipients to address the graduating class, unfortunately they're behind me, encouraging them to go forth and conquer the world. In the specific case of graduates of this great university, this means to go forth and make great biomedical discoveries. I have precisely the opposite advice to give you, dear graduates. In 4 words, my advice is go forth and fail. Laughter. Thank you, thank you, thank you. This advice is based on my own personal experience, which I shall now relate to you. 30 years ago, I was honored by being chosen for the very distinguished annual Flexner Award and lecture at the University of Pennsylvania School of Medicine. On the afternoon preceding the lecture, I was preparing my slides on a glass table in my living room. In those days, before you graduates were born, slides were mounted in cardboard frames. Some of the older amongst you may remember them. The frames were attached using a special kind of Italian glue. Having laid out all of my slides for my talk, I went upstairs for a couple of hours to make some telephone calls and to pack for my trip to Philadelphia. When I came downstairs to pick up my slides, I found that they had been chewed up by my Siberian husky. All of them. It turns out that Siberian huskies love Italian glue. I proceeded to Philadelphia without any slides. At my award lecture the following afternoon, the dean made a beautiful introduction of me and my work. It was not as beautiful as Mark's. I proceeded to the podium. After thanking the dean and the selection committee for this award, I said, "My dog ate my slides." The audience erupted in hilarious laughter. After they had calmed down, I went to say, no, I really, really mean it. The dog really did eat my slides. The laughter became even louder. So a few years ago at a scientific congress, a colleague of mine who had been at my Flexner lecture at Penn came up to me and said, "Your joke about your dog at the Flexner lecture was the funniest thing I've ever heard in my life." So, therefore, dear graduates, if you wish to avoid such traumatic experiences as I experienced in Philadelphia, my advice to you is to be sure not to make any important discoveries that might lead to important lectures at medical schools. Thank you once again, Mark, Russell, and the Honorary Degrees Committee for having recognized me.
Thank you, Paul. And would you please put on your hat? It's now my great pleasure to recognize Marilyn Harries Simons and James Harris Simons, cherished members of our campus community and two of the most exceptional philanthropists of our time. Marilyn and Jim have been extraordinarily generous, and it is no exaggeration to say that Rockefeller would not be the vibrant and scientifically robust institution it is today without them. They've helped us recruit world-class faculty, strengthened our programs at the interface of physics, mathematics, and biology, and sustained the outstanding clinical research programs at the Rockefeller Hospital. They provided critical, unrestricted support during the Great Recession, support that enabled the university to sustain its scientific excellence during a very challenging period in our history. Theirs has been institution-building philanthropy, and all of us in this great hall today have benefited from their vision and largesse. But what sets Jim and Marilyn apart from many benefactors is their deeply held belief in the importance of basic research. They understand that basic science requires patience, but in the long run, and sometimes in the short term, it pays huge dividends. Understanding the fundamental processes involved in life can have enormous implications for disease. Discoveries in one field may have value for insights into a completely different and seemingly unrelated field. Behind every pill and vaccine, behind every MRI and CAT scan, and behind every practical item that we take for granted, such as the radio and the internet, there was first new knowledge that paved the way. Jim and Marilyn know that the world needs individuals of extraordinary brilliance and curiosity who are given the time and support by enlightened institutions to pursue their questions, their hunches, their dreams, even their most improbable ideas. Marilyn and Jim see the wisdom in investing in the Faradays, the Ehrlichs, and the Merrifields of today. In this respect, they echo and renew the words spoken by John D. Rockefeller Jr. at the founding of the Rockefeller Institute for Medical Research. Famously, Mr. Rockefeller advised the Institute's scientific staff, I quote, "Don't be in a hurry to produce anything practical. If you don't, the next fellow will. You, here, explore and dream." I'd now like to say a few words about Marilyn Simons, who is one of the most dynamic people you'll ever meet, as well as one of the leading advocates in the United States for research on autism. Marilyn earned her bachelor's degree and her doctoral degree in economics from the State University of New York at Stony Brook, which is where she and Jim met. She is perhaps best known for her role as the president of the Simons Foundation, leading it through its formative period to its current position as one of America's most dynamic and creative foundations. It's no overstatement to say that the Simons Foundation has profoundly altered the face of American philanthropy. It provides extraordinary support for research in the basic sciences and mathematics, as well as in the life sciences. The foundation has a particular focus on investigations of autism and related disorders, and it has encouraged many of the world's great geneticists, biologists, and clinicians to include the search for the cause of autism in their research. The foundation has also created an invaluable database of clinical and genetic information about families affected by autism. Marilyn's leadership of the Simons Foundation is the capstone of a career dedicated to ensuring that children have the resources, environment, educational support, and love needed to maximize their potential and realize their dreams. This theme is manifest in her role as trustee of the East Harlem Tutorial Program, her leadership as a trustee of the Churchill School, a New York City school dedicated to helping children with learning disabilities, and most particularly, her devoted decade-long service as president of the board of the Learning Spring School, a private New York City school for children on the autism spectrum. In this last position, she oversaw the development of a model kindergarten through 8th grade curriculum. Here at Rockefeller, Marilyn has been an active member of the Women in Science Committee. Let me now turn to Marilyn's remarkable husband, Jim Simons, who was elected to the Rockefeller University board in 2000. He's been an exceptional trustee, helping to ensure the university's fiscal health through his service on the board's investment committee. Jim possesses one of the great intellects of our era. He's a man of many talents, a world-class mathematician, a phenomenally successful investor, and last, but perhaps not least, a champion poker player. Jim grew up in the Boston suburbs of Brookline. From a young age, he was a precocious mathematician who thought about numbers and shapes. He knew he wanted to do math from the age of 3, and he certainly got his wish. His passion for mathematics has influenced his entire life. Jim earned his bachelor's in mathematics from MIT and earned a Ph.D. in math from the University of California at Berkeley at age 23. He subsequently taught math at MIT and Harvard before joining the Institute for Defense Analysis, a Defense Department think tank in Princeton, where he worked as a cryptanalyst, breaking military codes during the Vietnam War. His next stop was the Stony Brook University, where he served as professor and chair of the mathematics department, building it into one of the world's top centers for geometry. During this period, Jim worked on a number of challenging math problems, and in 1975, he was awarded the American Mathematical Society Veblen Prize in Geometry for his work in differential geometry. Jim's most influential research involved the discovery and application of certain geometric measurements, now called Chern-Simons invariants, which have wide use, particularly in theoretical physics. Then, in the late 1970s, Jim left academia for the world of finance, starting Renaissance Technologies, one of the top-performing investment firms in the world. And the rest, as they say, is history. In addition to his service as a trustee of Rockefeller, Jim created the Math for America program, whose mission is to improve mathematics and science education in U.S. public secondary schools by building a core of outstanding STEM teachers and school leaders. He is chairman of the Simons Foundation and serves on the boards of many other prestigious institutions, including the Institute for Advanced Study, MIT, and Brookhaven National Laboratory, where he spearheaded the philanthropic effort that kept the relativistic Heavy Ion Collider operating. He is chairman emeritus of the Stony Brook Foundation. With these remarks, I provided the merest glimpse of Marilyn and Jim's accomplishments in advancing the frontiers of science. A full accounting would take many hours. Today, we're deeply privileged to salute Marilyn and Jim Simons for their many outstanding achievements and to celebrate their far-reaching service to our nation and to our world, conferring on them the Rockefeller University's highest honor, Doctor of Science, honoris causa. Marilyn, please rise to receive your hood and diploma.
President Tessier-Lavigne, Board of Trustees, distinguished faculty, I'm so moved to receive this honor, an honorary degree from Rockefeller University. It's such an outstanding research institution. I'm humbled but delighted to be here today. And congratulations to the class of 2013. I'm happy to join your proud families in celebrating your achievements today. My own educational background is very different from yours. I have a Ph.D. in economics. As a young girl, I went to Catholic girls' school on Long Island, where I remember one day the principal announced she was going to petition the Board of Regents to drop math and science from our curriculum because she didn't think girls should have to learn that. Fortunately, my science education didn't stop there. Through my work at the Foundation, I've been lucky to hear many outstanding scientists talk about their work with clarity and with enthusiasm that even I could understand as a non-scientist. Over the years, I've come to develop a love of science and an appreciation for the potential of research mathematics. But not everybody gets a second chance to learn about science. So at our foundation, we thought we'd expand our programs to try to offer more opportunities to enhance the public's understanding of science. About 6 months ago, we had this discussion, and I was particularly keen to expand our website content of math and science, hoping that we would syndicate this information and, disperse information to the public. We hired a very talented science journalist, and we agreed to editorial independence, and we looked forward to seeing our website visits rise. Our first article was launched a little while later, quietly, and staff told us Scientific America wanted to pick up this article for their website. While that was encouraging, internal reactions were not as positive. In fact, one scientist sent an email saying, "Remove that from our website immediately." He used words like flawed, premature, and crank in describing it, and he instructed the communications team to consult with him before posting articles on science. He didn't understand about editorial independence. This scenario was unfortunately repeated a little while later. We spent months struggling with journalistic independence, and by now we have a more nuanced understanding of terms like accuracy, errors, conflict of interest, human interest, advisory board, and cranks. Just last month, though, we had a wonderful experience. We published an article about the twin primes conjecture, and our website crashed, handling 70,000 visitors our first day. Reddit, Slate, and Wired also picked up our article, and I just heard it's being translated into Chinese and being disseminated in that language. But most exciting to us were not the website hits, but all the online comments we received in return. We heard from people who hated math but loved our article. People who complimented the author on clarity in writing science, and people who talked about the need for more good writing in science. My favorite comment was, "I read all the way through to the end with understanding. Either someone slipped me a smart pill this morning, or Erica's an outstanding journalist." In sum, our experience in enhancing the public's understanding of math and science has been stressful and educational, exhilarating and exhausting. We've learned there is indeed an audience for sophisticated science and math content. Also, outstanding scientists who are passionate about their work need to be part of the forum. And reliable resources for information are in high demand and low supply. That's from my economics. In our world, politicians and special interest groups influence many decisions critical to our country. We need the voice of scientists. As Alan Alda cautioned, maybe you all heard him when he was here, as he cautioned, if scientists can't communicate with the public, with policymakers, with one another, the future is going to be held back. So, class of 2013, you're behind me, but you have a bright future ahead of you. Many of you will use your education and imagination to excel in research. I'd like to suggest that you also use your talent to share your discoveries and your passion with others. Your creativity and hard work are an inspiration. Your sense of wonder and vision give us hope for a better world tomorrow, and sharing that is the most compelling message of all. Congratulations.
Thank you, Marilyn. And now, Jim, please rise to receive your hood and diploma.
You know, that woman started as my babysitter. She came to babysit our kids, and she's come a heck of a long way, it seems to me, from that time. So I'm very honored to receive this degree from such a wonderful institution. Now you're going to have to steel yourselves for a few minutes, because I'm going to give you a little lecture about mathematics. No formulas, no formulas. So, as Mark said, I was a mathematician, and in the early years I was a pure mathematician, coming up with concepts, proving theorems. Later, I applied mathematics to the world of finance, and that worked out too. But what was particularly interesting, as Mark already mentioned, some of my work became important in physics. Now, that was not through my plan. I didn't know any physics then, and I don't really know any physics now. But, and I was, of course, pleased to hear that. And, but I wasn't shocked. Even though I had no notion that this would apply to physics. The interaction between math and physics, the very strong interaction, has gone on since the time of Archimedes. And every physical principle is expressed in the language of mathematics, and sometimes the mathematics itself suggests new physics. I'll give you one example. I'll talk about an equation, but I won't say it. See? So Dirac, who was a great physicist, but very mathematically inclined, formulated in an elegant equation for the behavior of an electron. We all know what an electron is. And this equation was a new equation for the electron. And the only thing is, it had two solutions. It didn't have one, it had two. Now, one was described the electron. But what was this other one? Well, most people thought it was nothing. It was just an artifact of the math. But Dirac didn't necessarily feel that way, and neither did others. And within about a year and a half, the positron was discovered, and it fit exactly the bill of the other half, the other solution to that equation. And now we have PET scanners. So here was the mathematics which drove the creation of some new physics. So we see physics leading to math, math leading to new physics. The mystery of why mathematics seems always at the ready when physicists need it inspired a very famous article by Eugene Wigner, himself a famous physicist, entitled "The Unreasonable Effectiveness of Mathematics in the Natural Sciences." It's a great article, and if over the summer, you guys might want to dig it up and read it. So I asked the question, will there ever be a similar tight coupling between life science and mathematics? Now, I have to say at the outset, as a youngster, as Mark pointed out, I love math, and I hated biology. I couldn't make head or tail of it. It seemed to be just an endless stream of memorizing facts about flowers and frogs. None of it seemed to hang together. Darwin was never mentioned. It was just like, you know, learn all these things and try to remember them long enough for the test, which I wasn't very good. So I went for a number of years with not much of an impression of biology. But then I read The Selfish Gene, a famous book that many of you have heard of probably. And that gave a very good account of evolution. And while there were no numbers, it was coherent. So now I said, okay, there is something coherent about this biology that I was forced to learn or try to learn as a kid. Now, then I read The Double Helix, another famous book, and that made me realize, there's real mathematics that can apply to biology, at least in understanding the structure of complex molecules. So, okay, there's some math in biology. It makes some sense through evolution. Now, as our foundation got into the business of autism research, I was gratified to see that mathematics had found its way into many aspects of life science: genomics, proteomics, neuroscience, et cetera, et cetera. So, and quantitative biology is now a burgeoning field. And from personal experience, I can tell you that bioinformaticians earn a pretty good living, because I've tried to hire a few and succeeded, but paying more than I expected. Moreover, the sophistication of the mathematics being applied to biology has increased. Next week, a first-class biologist, accompanied by equally first-class mathematicians, are coming to our foundation to demonstrate an application of something called algebraic topology to understand certain biological data sets. Now, I don't know how many of you have ever heard of, anyone ever heard of algebraic topology? Raise your hand. Oh, we got 3. Okay. Well, algebraic topology, I mean, that's pretty hot stuff. Maybe mathematics is really getting somewhere, and this is all great. But here's a cautionary tale. So some years ago, I befriended a fellow named David Hirsch. Actually, I met him through Rockefeller. He's a well-known biochemist. In fact, he was chair of biochemistry at Columbia, and he invited me over to his lab one day at my request to show me what he was up to. Now, David's an expert on inflammation, and he showed me some work that he'd done on understanding the signaling pathway by which the body knows where to send blood to the area it's supposed to go to. You know, you scratch yourself, and a few moments later, it all turns red. So what's going on? How does that happen? So David explained to me. What a bananas mechanism. I mean, this cell communicates with that cell, which communicates with itself internally, and then signals to another cell, which goes through the same internal communication process, and then it signals to another cell. And it goes on. I mean, there were an awful lot of cells in this pathway. Finally, it got somewhere, and then there was another similar thing to tell it to stop with the, I mean, it was totally crazy. And I said, David, what theory could possibly have predicted so crazy a system? And to which he responded, no theory. It's just the randomness of evolution. Well, okay, maybe he's wrong. Maybe someday there will be some overarching mathematical approach to guide us in our quest to understand the biological universe. But even if he's right, and mathematics is never a full partner of life science the way it is in physical science, it will surely come to be its indispensable, loyal servant. It's just going to go more and more in being embedded in biology, and it behooves all of you setting out on this quest, you guys, and for that matter, some of you out there, to arm yourselves for the journey by learning as much mathematics as you possibly can. So go forth and learn math. That's it.
Thank you, Jim. Günter Blobel is one of the major figures in cell biology, a field that has undergone remarkable changes thanks to his fundamental insights. His research resolved a basic problem in cellular organization. How are the proteins in a human cell, which may number 1 billion individual molecules, distributed to distinct cellular addresses? Gunter, who is renowned as an intrepid explorer of nature's inner world, delved into the challenging questions, these challenging questions at full force when he came to the Rockefeller University back in 1967. His discovery that proteins contain built-in signal segments to orchestrate the traffic has greatly expanded our understanding of normal cellular functions as well as several inherited diseases, and it has informed drug development as well. Gunter was born in Waltersdorf, Germany, now a part of Poland. In January 1945, he and his family fled the advancing Red Army and found themselves near Dresden, a cultural center often described as Florence on the Elbe. A month later, Gunter witnessed firsthand the Allied firebombing of Dresden, the intensity of which he later described as giving off enough light to read a newspaper even though it was the middle of the night. Gunter attended the University of Tübingen, receiving his M.D. in 1960. He moved across the Atlantic to the University of Wisconsin-Madison, where his oldest brother, Hans, was a professor. Gunter joined Van R. Potter's group in the McArdle Laboratory for Cancer Research, and he received his Ph.D. in 1967. From there, as a postdoc, he joined the laboratory of Philip Sikowitz and George Palade at Rockefeller, and he's been here ever since. Palade and Sikowitz were studying the functions of various organelles in animal cells. In order to keep separate the many processes carried out in the cell, organelles have impermeable membranes. So the question of how a protein reaches its assigned organelle came with a corollary. Once a protein reaches its destination, how does it gain entry? Gunter focused on the endoplasmic reticulum, a system of membranes that assist in the genetic translation carried out by ribosomes. The endoplasmic reticulum also is responsible for preparing secretory proteins for their journey to the cell membrane and beyond into the bloodstream. In 1971, Gunter hypothesized that eukaryotic proteins contain zip code-like signals in their molecular structure that act as address tags to guide them to the part of the cell to which they're assigned. The destination could be a specific organelle, a location on the cell membrane, or the bloodstream. He dubbed this concept the signal hypothesis. In 1975, Gunter developed a unique cell-free experimental system designed to simulate the processes of protein translation and translocation, which allowed him to break these processes down and observe their individual biochemical steps. By 1980, he demonstrated the general molecular principles underlying this process. Ultimately, he showed that the coding system he had hypothesized comes in the form of signal peptide sequences attached to each protein, and that these peptide sequences act not only as address tags, but as a kind of cellular global positioning system that guides the protein to a specific location, and also as a key to gain access beyond the otherwise impermeable membrane. Gunter has been honored with many awards, including the Albert Lasker Award in Basic Medical Research and the Nobel Prize in Physiology or Medicine. Just as Gunter has been a leader in shaping the landscape of molecular cell biology, he's also had a leading role in restoring the urban and cultural landscape of Dresden, Germany. In 1994, Gunter founded the Friends of Dresden, a philanthropy committed to the reconstruction, restoration, and preservation of monuments and buildings of architectural and historic significance that were destroyed by the Allied bombing in World War II. Dresden's Frauenkirche, a magnificent church where Bach once played, had made an indelible impression on Gunter when he first saw it in his boyhood. In 1999, when Gunter was awarded the Nobel Prize, he donated the entire monetary amount of his prize to help support the rebuilding of the Frauenkirche and the building of a new synagogue in Dresden. Gunter's contribution was the single largest individual donation to the project, and it galvanized countless others to join him in giving. Gunter and his wife, Laura Mioglio Blobel, have also donated a small portion of the monetary prize to restore an old Baroque church in the hometown of Laura's father in Italy. Gunter has been a pioneer and an inspiration through his scientific discoveries, through his mentoring, and through his philanthropic efforts in Dresden and beyond. We are greatly in his debt, and we are delighted to have the opportunity to recognize him here today. Gunter, please rise to receive your hood and diploma.
Chairman Russell Carson, President Marc Tessier-Lavigne, graduates and honorary graduates, ladies and gentlemen. First of all, thank you, Marc, for your very kind and extensive laudatio. I would like to congratulate the class of 2013 on their PhD degrees and their parents and families who rightly and richly deserve to feel the pride and the joy of this occasion. For my part, I was honored to share this stage with you and the distinguished honorary degrees recipients, Drs. Marilyn and James Simon, and my colleague, Professor Paul Greengard. Would I have any pearls of wisdom to dispense on that occasion, based on my experience, may be useful for your own career, or nice, inspiring jokes to tell? Of course, many. But being the last speaker and seeing that the orchestra is already here for the exit, I decided to whittle down my long list of wisdoms, most of which are obviously personal and therefore of limited relevance. Not even for those of the 2013 graduates who want to follow a career as a research scientist. But nevertheless, here's a very short list. Keep a child's curiosity. Jim Simon has just told us how he kept his curiosity and became interested in biology. Don't lose this child's sense of wonderment. If you do that, then choose to do something else, but research. Learn through teaching. If your grandmother doesn't understand what you are doing, you probably don't understand either. Invite criticisms and accept opposition to your ideas. But trust your instincts and stay the course. And in closing, here is one of my favorite jokes. I promise you at least one joke. And this joke has unfortunately gone into the internet. You are one, I told many of my students, and they are many. And if you don't work day and night, you will be crushed like a roach. Of course, this is not a very good way of ending. But anyway, I'm very grateful that the selection committee chose to give me an honorary degree. And again, I congratulate all of the newly minted graduates and wish you the best for your future. Thank you.
Thank you, Gunter. And now, Paul, Jim, Marilyn, and Gunter, I would ask you to please rise. By the authority of the Regents of the University of the State of New York, which they vested in the trustees of the Rockefeller University and is by them delegated to me, I confer upon you the degree of Doctor of Science, honoris causa, which admits you to all the rights and privileges thereunto pertaining. Congratulations. Please be seated. Paul, Jim, Marilyn, and Gunter, Chairman Russ Carson, graduates, faculty, staff, and students, this concludes the 55th Convocation of the Rockefeller University. Now, I ask that you all please rise.